All right, welcome back. New video. It's my third one that I've made tonight, so hopefully everything's going well with you guys. All right, let's get into a few more things. We kind of just barely got an intro going. I'd like to get a little further into the material. Again, most of it's just introduction right now. We're trying to get to a point where we're competent and we can do more fun things like case studies and and analysis and deciphering what type of bacteria is which. Um, and talk more about pathogens and such. So we'll get there, but first we got to kind of establish the bare minimum. All right. So let's look at some reasons why we're looking at microorganisms and kind of go from there. So microorganisms have a profound influence on all aspects of Earth and its residents. Bacterial-like organisms in the fossil record as far back as 3.8 billion years ago. So these are prokaryotes, organisms without a true nucleus. Right, we do have record of certain forms of algae going back quite a bit as well to really early stages. So yeah, we definitely see certain bacterial blooms and evidence of those from that long ago. All right, around 2 billion years ago, we get eukaryotes, organisms with a true nucleus. All right, so now we have an enveloped nucleus and quite a bit, which is going to add a decent amount more complexity because we're going to have to control what comes out in and out of the nucleus, but it's also going to have to worry about breaking it down during replication. And but to the advantage of the eukaryotes, because they have their organelles encased in membranes, there's a lot more protection, right? Especially nucleus to DNA, right? So there are advantages to having a nucleus. Right? But there are advantages to prokaryotes, right? Such as reproduction. They reproduce very quickly, right? All right. Bacteria appeared approximately 3.8 billion years ago. Okay. Origin of the Earth. This is the number that keeps getting revised, right? We're going off of certain cosmic background radiation. We're going off of the decay of certain elements in the Earth, elements that had to be there, had to be formed in the explosion of the star that had the matter that formed the Earth. We know that certain of those rare earth elements, such as uranium, can only be produced in that nuclear explosion that occurred when a star supernovaed. So that, but we can track the decay to come back and determine, hey, this is decayed for so long. How old is the Earth? Right now, we're getting somewhere. This is showing about 4.5. So somewhere between 4.5 and 4 billion is about what we think now. Last time I think I saw it was like 4.3 or so. So that's the probable origin of the Earth. And then we get life, prokaryotes appearing somewhere in that 3.8 billion years, and then prokaryotes dominating, right? So those ancestors to the archaebacteria and eubacteria there, and we don't really get eukaryotes here until right here, less than 2 billion years ago, and then we get to a billion years ago, and then we get reptiles appearing. You know, what is that? 700 million years ago. Cockroaches, termites, right? So we're getting into our insects here. Some of our more advanced insects a little bit later there. Mammals appearing, humans appearing, right? When you think about how long we've been here, it's not very long, although considerable impact upon the world in our existence, right? All right, microorganisms are everywhere. I've talked several times about our microbiome. We need those bacteria. We need them to be happy. It helps. It helps with our health. They occur in large numbers, and they live in places many other organisms cannot. We find archaebacteria, the extremophiles, in really cold and freezing environments. We find the archaebacteria in vents along volcanoes. We find archaebacteria down in the deep ocean vents. So we do find these bacteria in places where we think no other life could occur. which makes sense in that bacteria being that ultimate originating organism, right? Or the ancestor there too. All right. Microbial involvement in energy and nutrient flow. So bacteria conducted photosynthesis before we even had plants, right? So bacteria had the ability to take the sunlight in and move those electrons and redox reactions in order to harness some energy. In fact, when we think about plants and we think about animals, we have to think about uh, endosymbiotic theory, where we have some eukaryotic ancestor that likely engulfed a bacteria, and that ended up becoming the mitochondria, or engulfed a prokaryotic organism that was able to conduct photosynthesis, and that became right the various membranes involved in the chloroplast. So we definitely have those know that bacteria played a role even in the evolution of eukaryotic organisms. Right. So we had anaoxygenic photosynthesis, the ability to get run photosynthesis without oxygen and then with oxygen, which is really what we see now, right? In plants. Biological decomposition and nutrient recycling. So bacteria break things down, right, really well. We know one of the things, we don't want to touch dead things because of bacteria. That could be on those dying things that are breaking it down, bacteria that we don't want. In fact, that could come back into play in some case studies we look at in the future. And in nutrient recycling, right, by breaking things down, they're breaking apart those different things like nucleic acids and carbohydrates, and they're freeing up these elements so that they can be reused, go back into the earth and be reused. All right. Now, the great news is that we can use microorganisms to a lot of our own ends, right? Humans have been using microorganisms for thousands of years. Bakers and brewers use yeast, right? In order to, brewers, especially for fermentation, right? We got to get that fermentation to produce the right amount of sugar. Bakers, we're going to need, or to break down the sugar in fermentation, right? And then for bakers, what we're going to need yeast in order to produce the carbon dioxide to get things to rise and rise to the correct amount. Cheeses, right? Cheeses are fermented, and mold plays a role in some of them, right? So a lot of our dairy products, we're concerned with microorganisms there, right? And using things like moldy bread on wounds, using the mold to clean out other organisms, right? We can also use these for certain things like oil biodegradation. We can use some of these microbes and take over their genetic codes to produce things we want, such as hormones. Like we can use microorganisms to make more of insulin that's badly needed, so that we don't have to take insulin from the glands of cattle that are being slaughtered. So there are some advantages there as far as the idea that some organisms are more valuable than other organisms, right? All right, biotechnology and bioremediation, right? Biotechnology, when humans manipulate microorganisms, which is kind of what I was just talking about, to make products in an industrial setting. Genetic engineering, so what I was just talking about was making genetically modified or bacteria. A lot of times, I mean, when you look at the human growth hormone epidemic that engulfed baseball in the early 2000s, that went back to a lab that was using bacteria to manufacture that human growth hormone. They were hijacking the bacteria to make that human growth hormone in order to make that industry work. Recombinant DNA technology used to engineer GMOs capable of synthesizing desirable proteins. So like I said, we can use them to make proteins, medicines, hormones, and different enzymes that people may need to take as therapy. All right. Bioremediation, we can use the activity of microbes to clean up the environment, to help break down the chemicals in oils for oil spills, for different other chemical spills. We use microorganisms all the time in water and sewage treatment to make sure that we break down the things that we don't want in the sewage. All right. Okay, so pathogens are just simply disease-causing agents. And when we start looking at top causes of death and diseases, right? Heart disease is still the number one killer in the United States and worldwide as well. Okay. Cancer is next. Then we have stroke. And then we start getting into certain things where infections come into play, right? We see respiratory infections, we see influenza and pneumonia, HIV/AIDS, diarrheal diseases. We'll talk about several of those coming up, such as cholera being a big one that we have largely under control in developed nations, but in developing nations can cause major issues, especially when a well in a village gets Vibrio cholerae in it, which is a bacteria that loves water, and then you got a whole village where everyone's sick and dying. Septicemia, right? That's a infection that's in your bloodstream, bacterial infection of your bloodstream, and that's really bad because now it can spread all throughout your entire system, right? Influenza and pneumonia, a lot of people don't think about it, but pneumonia, especially among women in their 40s and 50s, is a large killer. My mom actually had a really bad case of pneumonia, didn't realize it, thought it was the flu, and she had to be put into a medically induced coma for about 2 or 3 weeks in order to recover. And we were worried about all kinds of things because the antibiotics they gave her ended up causing some issues, and we had to control some of the fungi that were causing some issues as well. So it was quite an ordeal. Luckily, my mom pulled through and is alive today. But it just kind of reminds you of how quickly things can turn and how important it is to know about these different diseases. In fact, my mom had been to the doctor a few days earlier and they said flu, and then she was barely functioning, barely able, and my dad and I took her to the hospital, and then they care flighted her to a even better hospital. So always important to make sure you know if things aren't getting better, do go back to the doctor, right? Okay. Tuberculosis, well, again, another thing that in the developed world we have largely under control. Tuberculosis and consumption, right, is one of those major things that was a big concern even here in America in the 19th and early 20th centuries, and is still a big issue in a lot of developing countries. All right, so kind of looking at the most common infectious diseases worldwide, right? We got some ones that we know pretty well, like AIDS. We got ones, respiratory infections. Those can be viral and bacterial. Influenza caused by the flu. Pneumonia can be caused by bacterial or flu. A lot of times it just has to do with fluid buildup in the lungs. Get diarrheal diseases. Cholera, dysentery, typhoid, some really important ones there. Tuberculosis, malaria, a big one we're concerned about, and the idea behind mosquitoes and gene drive. Hopefully we'll get a chance to explore and talk about later in the year. Measles, hepatitis B, tetanus, parasitic diseases, and some miscellaneous. Now, some of these we might know. We have pretty good vaccinations for measles. It's just getting those vaccinations everywhere around the world to where people actually need them, or people just not getting vaccinations. That plays a role there. Hepatitis B is also preventable. We also have vaccinations for hepatitis B. In fact, a lot of you may have been vaccinated for it or will get vaccinated for it before you go off to college. Same thing with tetanus, right? With that DPT vaccine, right? So we do have a lot of different things that can be dealt with, but it's just about getting those vaccines to everybody and making sure those are shared equitably. It's hard when when we're talking about pharmaceutical markets and capitalism. There, it's hard to think about like how can these overlap? How can we make pharmaceuticals but still be equitable in how we share them out? And it was interesting to see that a lot of the people making the COVID-19 vaccine did vow to try to be more equitable with who gets the vaccine. But then the immediate question that comes out ethically is, what does equitable mean, because we're talking about corporations, pharmaceutical corporations that haven't always shown to distribute these equitably. All right. Okay, so increasing number of emerging diseases are around. We can add COVID, which is SARS-like virus, coronavirus, right? SARS, if you remember that outbreak from the early 2000s, AIDS, hepatitis C, viral encephalitis are several common ones. Other diseases, and I would add Ebola virus on there as well. One that sparks up here and there. Marburg virus would be another interesting one to look at as well. Other diseases previously not linked to microorganisms now are. Gastric ulcers are all about the Heliobacter pylori bacteria. Certain cancers are linked to certain viruses, such as the HPV, right, the human papilloma virus, for right, that can concussional genital warts and so on, schizophrenia, multiple sclerosis, obsessive compulsive disorder, coronary artery disease. So we've got a list of these things growing where we're seeing more and more microorganisms are having some kind of effect on these. And so now we can think, oh, how can we prevent them from getting into those places? What can we do and treat these bacteria? Or is the damage done? Is there something we can do preventative before the damage gets done, right? All right. The other thing we're increasingly worried about, and something we'll look at, is drug-resistant strains. A lot more types of bacteria are becoming drug resistant. They're able to transport the drugs right outside of them, right. They'll be able to. They have these ABC transporters, which they can use to package up the drug and ship it out before it can do its damage. So we'll look more and more at that. Interesting to know. We think back to penicillin kind of being one of those wonder drugs coming out of the 40s and early 50s, and it had an immediate effect as an antibiotic, and a lot less people were allergic to penicillin than they were to the sulfa drugs that were used before. In fact, I'm allergic to sulfa drugs, so I'm so glad that things like penicillin were invented. I'm not allergic to penicillin. Some of you may be, but there's probably other antibiotics you aren't. But they saw that within 2 years of the widespread development of penicillin, they had already found bacterial resistance to penicillin. So we knew pretty quick that this was going to be an issue. And part of the problem we have, I think, is that we flood antibiotics into everything. And you might have heard about antibiotics being placed in chicken feed, right? Just to try and keep more chickens going into production. But you kind of worry the more and more those antibiotics are out there, and the more and more strains that can develop ways to deal with those antibiotics, right? Life finds a way, right? Okay. We mentioned prokaryotic versus eukaryotic already. Let's give a few differences here. Prokaryotic, right, organisms, bacterial and archaeal cells, are about 10 times smaller than eukaryotic cells. Prokaryotic cells lack many cell structures, such as double membrane-bound organelles. All prokaryotes are microorganisms, but only some eukaryotes are. Right? When we think about eukaryotes being microorganisms, we generally consider certain algae that are, and what we consider protists would fall within those microorganisms. Yeasts, which are single-celled, would also fall in there as well. All right, there's a difference between the cell structure of a prokaryote and eukaryote. Viruses are neither but are considered particles. So we talk about viral particles. So for cells, right, here we got a typical prokaryotic cell. We've got a cell wall and a cell membrane. So cell wall, cell membrane. And then within the cell, we have ribosomes, right? There's no membrane-bound organelles, where ribosomes are there in order to produce proteins from mRNA, which is being sent from they don't have a nucleus, from the chromosome. So, and what we typically see in bacteria is we see circular chromosomes. So what they're showing you here is a circular chromosome here that would contain the genetic code of the bacteria. And so what you have there was kind of awesome to me about bacteria is the second an mRNA is being made, right as it's being transcribed from the DNA, ribosomes immediately latch on to the sections that have already been processed and immediately start forming proteins. Whereas for eukaryotes, we got to find a way to get the mRNA out, processed incorrectly out of the nucleus before it can even get to the ribosomes in order for proteins to be made. Now, in eukaryotes, that can be advantageous because we can there's various checks that can be done to make for the integrity of the mRNA, whereas with prokaryotes you don't have that. But what's great about prokaryotes is that they don't live very long. And you might think, oh, that's a disadvantage, but they reproduce quickly, which means that they can that some certain mutations may not always be bad, or there's lots of them. So even if you have some negative mutations, some negative outcomes from some proteins that don't work due to some errors in the mRNA, it's not the end of the world as far as just losing one bacteria when there's millions being made around in the environment. We talked about a lot of times there's lots of them where you find them. All right, and then over here we have a virus, right? Viruses are tiny particles, not cells. Usually there's some kind of capsid or envelope, right? So the capsid here would contain the DNA, and then it's able to transfer that DNA into its host cell. What we're looking at here is a bacteriophage. Bacteriophage is generally, we're looking at infecting bacteria. Here we're looking at an enveloped virus where we get a viral envelope. That would be like COVID, like the SARS-CoV-2 virus, which produces COVID-19 disease. All right. So here's the envelope, right? And then we have the spike proteins on the outside that are able to recognize by cellular receptors, which allow the viral particle to be allowed into the cell for the genetic material. Then once your genetic material is in the cell, boom, that's when everything starts happening. Okay. And we'll talk more about that when we get to viruses. All right. There are 6 main types of microbes. So bacterium or archaeon. So that's what we're talking about with eubacteria and archaebacteria. We got fungus or fungi. We got alga, which I mentioned a few times differently. With the alga, what do we have? Well, we have the cell. We have within these cells chloroplasts, right? Within these single cells, chloroplasts. Viruses, protozoans, and we'll talk a little bit about helminths, which is getting into certain types of worms. Okay, so we'll try and get into some worms. We'll try and get into some segmented worms, or some flatworms and some roundworms. I don't want to say segmented worms. I don't think we're going to really deal with annelids, but we'll get into some platyhelminthes and some nematodes when we get a chance. So I see some different ones. Notice the protozoans there typically are single-celled. Right. Here we see an enveloped virus. Okay. Here we see the bacteria in their environment. Here we see a type of fungus. All right, so we'll get through viruses and we'll call it today. Viruses are not independently living cellular organisms. They're much simpler than cells. Basically, there's a small amount of DNA or RNA wrapped in protein and sometimes by an additional lipid membrane. Individuals are called a virus particle or virion. They depend on the infective cell's machinery to multiply and disperse. And what we mean by machinery, they're going to hijack things. Some of these are capable of taking RNA, right, converting it into DNA using a reverse transcriptase, and then inserting it into your own genome. And now you got that viral DNA within a cell, and until that cell dies, that's always going to be part of that cell. We see some that are able just to immediately hijack the ribosome, and then just use the ribosome to transcribe the genetic material there. That's ready to go because they're already carrying the materials that they need. So boom, all of a sudden now the ribosome only is transcribing, or the ribosome's only making proteins from the viral material. So we'll look more and more at that. I find that part interesting. Obviously, being one of the people that co-developed the cell biology class with Mr. Tran, cells are near and dear. All right. So microorganisms vary in size from 1 micrometer to 200 nanometers. So remember, 1 micrometer is the same thing as 10 to the -6 meters, and 200 nanometers would be 2 times 10 to the -7th meters. So that kind of gives us a nice size there. We will get into certain vectors, like here we have a louse, or what we call lice when there's multiples. So we'll get into some things where organisms are spread through different vectors. Here we're kind of seeing red blood cell is here at 10 micrometers, right? Here's a colonial alga, so where different algae are living together. White blood cell, amoebas. So now we're getting to some protozoans that are here. Now we're getting to bacteria, right? So most bacteria are right here around that 1 micrometer or smaller. Okay. Some like mycoplasmas are even smaller and have some interesting membrane issues. Here we got some larger viruses, and there are some pretty large viruses. There's some megaviruses that are insanely large and incredibly rare as well. All right, and then we get down here and we're at 1 nanometer, and that's the diameter of DNA. Okay. Get down here, hey, here's a single amino acid. Okay. Hydrogen atoms down here at 1 angstrom. All right, so we'll stop here at lifestyles of microorganisms. I'll probably come back with a video pretty quick, maybe even a second one for the weekend, and we'll talk more about what we want to do in class. Y'all have a wonderful rest of whatever time you're watching this, and I'm signing off.